Okay, the last thing we're going to talk about is what is the human role in genetic engineering in regards to agricultural and medicine. So this is our last learning target for our heredity unit. You're going to be able to explain how human technology and activities can alter systems of heredity and interactions which could impact society for future generations. So we're going to be talking about genetic engineering and whether or not this is a good thing or a bad thing. Okay, so first of all, we need to figure out what is genetic engineering. So this is when there is a manipulation of an organism's genes using biotechnology. And when I put zero, there's no zero risk, meaning there's always going to be a risk when this is done. Okay, so a couple ways that this can be done. New DNA may be inserted into the cells, or you can remove genes. GMO stands for genetically modified organisms, so anything that's been genetically modified is called a GMO. And genetic engineering is used in medicine, agriculture, environment, and food production. Okay, another way that we have genetically engineered living things is cloning. Scientists working on the Human Genome Project to map every gene of a person and see what it does. They're not finished. As of right now, we have about 30,000 um, genes. Pretty much with cloning, what they do is they take an identical copy of the parent and they copy the DNA. It's used a lot in horticulture with plants. They actually did do it with an animal, Dolly the sheep. Okay, and you've actually seen this if you've watched the movie Jurassic Park. It is fiction, it's not real, but scientists have dreams of cloning extinct or endangered species. So let's take a look at Dolly. Okay, so the organism to be cloned, okay, so they take the cells, okay, from the organisms. They've been starved of nutrients so that they don't begin to make copies, so they don't go through interphase and mitosis. Okay, they copy the chromosomes right before the cells reproduce themselves. Two copies of the chromosomes can cause defects in an organism. So then the egg and cell to be cloned are fused together. And then once that egg and the cell that are going to be cloned are fused together, okay, then that cell becomes what we call fertilized, okay, and it can start making a um, a copy or a clone. Okay, so that's cloning. So let's talk about when genetic engineering is used. It's used in medicine. So you can produce large quantities called recombinant DNA of hormones, immune system proteins, blood clots, and cell proteins. So I'm going to have this linked to the notes. If you want to read a little bit more about recombinant DNA, this website will be available to you. It gives you a little bit more complex information about how recombinant DNA works. Okay. All right. So what recombinant DNA is it's basically taking a gene out of a cell, out of a chromosome of a cell, and put it into somebody else's cell so that that gene can work and help that person become healthy. So an example of this is with your pancreas. Inside your pancreas, that's where the production of insulin is created inside those cells. So what they do is they isolate the insulin gene, the gene in the pancreas that makes insulin. And they take that gene and they put it inside a bacteria called E. coli, okay, and they insert that, okay, into cells of a pancreas that does not produce very good insulin. And so people with diabetes, they're working on this to cure and treat people who have diabetes. So this is an example of how they can use pieces of DNA to heal or cure and create medicines for people with diseases. Okay, we also use genetic engineering in food. Okay, so rice, potatoes. Basically, the reason why we do this is we want higher crop yields, more nutrition. We want to be able to grow foods in harsh environments that normally can't grow as many foods. We want these foods to be resistant to disease, so we don't have to use pesticides, which is actually a good thing. And we also want to get rid of undesirable characteristics. And we want to have a longer shelf and flavor life. So what they do is they look for genes that can do all these targets. And that's what they'll do in search, or they'll breed only those types of plants that don't have any undesirable characteristics or traits. Problem with this is if they insert genes like they did with, like I showed you with the recombinant DNA, sometimes the gene can land in the wrong spot. So sometimes it doesn't go to the right chromosome. And this can cause harm to the organism or the living thing. And another negative part to this is that genetic modification, when passed on to offspring, that can sometimes harm the ecosystem. So remember, ecosystems a delicate balance between a food chain into a food web. And if you change one living thing in that ecosystem, and you maybe make it so that it's able to grow in harsh environments, well, you're adding a food source into an ecosystem that normally wasn't there. That might mess some things up. This could also cause allergies. It's been known whenever we genetically modify food, sometimes that can create allergies. So those are just a couple ways that genetically altering food can be a problem. Okay, so what I've done is just gone over what genetic engineering is, a couple different ways that you can genetically engineer food and medicine, and then talked a little bit about what we use it for in agricultural and medicine. You will be doing a lot of practice in a project with this target, and you'll actually be researching this last target, this last part of the target, which is talking about how genetic engineering is altering society and whether or not it should be continued.